how does the depolarization of the sarcolemma actually lead to muscle contraction? This is what we call excitation, contraction, coupling, or EC coupling. Remember that at the axon terminal, acetylcholine is released. Acetylcholine binds to the acetylcholine receptors that are chemically gated sodium channels. They open up, sodium goes in, causes a localized depolarization called the end plate potential. If that depolarization reaches threshold, an action potential is generated which travels across the sarcolemma, and that is what we call excitation. Now, we need to make that excitation actually cause the muscle to contract. This is going to occur when the action potential propagates down the T tubules, which remember are these infoldings of the sarcolemma that go down inside the cell. The action potential travels along these T tubules where it is in close contact with the sarcoplasmic reticulum. And the sarcoplasmic reticulum is then going to release calcium. And calcium is the actual signal for contraction. Note, the actual action potential is very, very quick, lasts very, very short amount of time, and it's actually over with before we actually see the muscle contract. We've already gone through the whole depolarization, repolarization before the muscle actually contracts. How does calcium lead to muscle contraction? Well, remember that we have the thin filaments and the thick filaments. The thick filaments are a protein called myosin, and myosin has these head groups on them that can grab onto actin. However, when there's a low amount of calcium inside of the sarcoplasm, the cytoplasm inside of the muscle cell, there is a protein called tropomyosin that's bound to actin that blocks the myosin binding sites on the actin. So when there's low amounts of calcium, the myosin head groups can't grab onto the actin because of the tropomyosin. Because of this, we have no cross bridges, so the muscle fiber is relaxed. The two fibers are not attached to each other. We have no cross bridging. When the action potential spreads across the sarcolemma and travels down into these T tubules, it's going to trigger these voltage-sensitive proteins. The voltage-sensitive proteins on the T tubule are going to activate proteins on the sarcoplasmic reticulum in these terminal cisterns that's going to cause the release of calcium ions into the cytosol. When the level of calcium in the cytosol increases, the calcium is going to bind to troponin. Troponin is then actually going to move tropomyosin out of the way. When troponin moves tropomyosin out of the way, myosin can bind to actin, and we get cross bridges forming. And then we are going to have this whole what we call cross bridge cycling, where the myosin is going to grab onto the actin, pull it, break the cross bridge, grab onto the actin again, pull it again, grab it again, pull it again, grab it again, pull it again, sort of like pulling a rope hand over hand, and we get the sliding of the filaments, which is going to shorten the sarcomere, and the actions of all of those sarcomeres shortening is going to shorten the overall muscle fiber, and the combined action of all of those muscle fibers shortening is going to shorten the muscle, causing contraction and generating force. Once that nervous stimulation ceases, because again, remember that enzyme acetylcholine esterase is going to break down the acetylcholine, causing the sodium channels to close back up. The calcium ends up getting pumped back into the sarcoplasmic reticulum, and then troponin is going to move tropomyosin back over the myosin binding sites, and contraction ends. The muscle relaxes. Once we have that calcium inside of the cell released from the sarcoplasmic reticulum, it is going to bind to troponin. The troponin moves the tropomyosin out of the way, and then we get the cross-bridge cycling. The cross-bridge cycling is going to occur in 4 steps. 1, we are going to have the myosin head group attached to the actin. And at this point, the myosin head group is already in what's called a cocked position. It binds to the actin, and then ADP and a phosphate are going to be released from the myosin head group in what we call a power stroke, where the head group actually pivots and pulls the actin filament towards the M line. So remember, the M line is this center line where all of the thick filaments are attached. So thin filament gets pulled. Then, in order to detach the head group from the actin filament, ATP actually needs to bind here. So ATP binds and it detaches. Then the ATP gets hydrolyzed. So it gets converted into ADP and a phosphate. And this process, remember, the hydrolysis of ATP releases energy. This release of energy is going to cock the myosin head group. So it's going to put it into this high energy state. So then, once it binds to the actin, it has the energy necessary to pull the actin, releasing the ADP and the phosphate, and it is ready to be reset by an ATP binding again. ATP is hydrolyzed, cocking the head group, and then it is ready to bind to the actin again, pull the actin, ATP binds, it resets. And it keeps doing this over and over again as long as the myosin is able to bind to the actin. And that's going to be true as long as there's calcium. However, once the stimulation of the muscle cell stops, it is going to have the calcium lowered because the sarcoplasmic reticulum sucks the calcium back up. Troponin moves tropomyosin back into the way, and we can no longer form these cross bridges, and the muscle relaxes. So, cross bridge formation. Myosin head group attaches to actin. Power or working stroke. The ADP and the phosphate are released from the myosin head group, and it pulls the actin filament. This is where we get the actual sliding of the filaments. Then ATP is going to attach to the myosin head group, and the ATP attaching actually triggers the myosin head group to let go of the actin filament. This way it is ready to go again. The ATP gets hydrolyzed, converted into an ADP and a phosphate, cocking the head group, putting it into the high energy state where it can grab onto the actin, perform a power stroke, detach when ATP attaches, hydrolyze the ATP, cock, grab, pull, ATP, hydrolyze, cock, grab, pull, ATP attach, and it detaches, hydrolyze, cock, grab, pull. Over and over and over again, this happens until we pull the calcium back out of the cytosol using pumps on the sarcoplasmic reticulum, troponin moves tropomyosin back into the way, and the head group is no longer able to attach to the actin. In rigor mortis, this is stiffness that we see in death. It occurs 3 to 4 hours after death. Muscles stiffen up, they contract. This is going to be due to the fact that we need ATP in order to actually detach the myosin head groups from the actin. So in order to break those cross bridges, ATP needs to attach to the myosin. After death, the cell no longer is able to regulate intracellular calcium levels. So calcium starts to build up inside of the cell. That is going to cause troponin to move tropomyosin out of the way, and then all of those cocked and ready myosin head groups are going to attach to the actin and perform a power stroke, releasing the ADP and the phosphate. But in order to break that cross bridge, ATP needs to attach. But because the cells are dead, they're no longer making ATP. So they don't detach, so the muscles stiffen up. And we call that rigor mortis. It starts 3 to 4 hours after death. We get peak stiffness about 12 hours after death. And this can be used to estimate time of death in crime scenes. The muscles are going to stay contracted and rigid until the muscle proteins actually start to break down, at which point the muscles are going to relax again. So just a quick overview of our big picture. 4 steps in order for skeletal muscles to contract. We need events at the neuromuscular junction. We need the muscle fiber to excite, produce that action potential. We need to have the excitation contraction coupling. And then we need the cross-bridge cycling. So at the neuromuscular junction, acetylcholine is released from the axon terminal. Acetylcholine binds to acetylcholine receptors. Acetylcholine receptors open. Sodium enters the cell, depolarizes the cell, makes it less negative. If that depolarization at the end plate potential reaches threshold, that triggers an action potential by opening up voltage-gated sodium channels. That action potential travels along the sarcolemma, causing more voltage-gated sodium channels to open. That change in voltage travels down the T-tubules, where it is going to change the shape of these proteins in the T-tubules. That is then going to cause the opening of calcium channels on the sarcoplasmic reticulum. Calcium gets released into the cell. Calcium binds to troponin. Troponin moves tropomyosin out of the way, allowing myosin to bind to actin, and then we are going to get the cross-bridge cycling that's actually going to pull the thin filaments past the thick filaments, shorten the sarcomere, and cause the muscle to contract.